Hello everyone, my name is Deepa Bajeta. I am working as an associate professor in IIMT College of Pharmacy. Today I am going to discuss about the topic carbohydrate metabolism and biological oxidation. So in this I will cover the content, carbohydrate metabolism, glycolysis, bioenergetics of glycolysis, TCA cycle, bioenergetics of TCA cycle, gluconeogenesis, significance of gluconeogenesis. So first is carbohydrate metabolism. The carbohydrates are the major source of energy for the living cells. Glucose is the central molecule in carbohydrate metabolism since all the major pathways of carbohydrate metabolism are connected with it. Glucose is synthesized from non-carbohydrate precursors and carbohydrate precursors and stored in the form of glycogen in our body to maintain the glucose blood level and when it's needed. It's also known as EMP pathway. So first is glycolysis. Glycolysis is the sequence of 10 enzyme catalyzed reaction that converts glucose into pyruvate with simultaneous production on ATP. In this oxidative process, 1 mole of glucose is partially oxidized to 2 moles of pyruvate. It is also known as EMP pathway, Amidan-Mirov-Parnas pathway. Sorry. Location. The main location of glycolysis occur is cytosol of all cell. It is unique pathway. It occurs aerobically as well as anaerobically. Inaerobically, presence of oxygen, glucose is converted into 2 moles of pyruvate, and in anaerobically, in the absence of oxygen, pyruvate is further converted in glucose. So reverse pathway of glycolysis that is called gluconeogenesis. It is also includes formation of lactate from pyruvate. In anaerobic organism, glycolysis is the prelude to citric acid cycle and electron transport chain. It is central pathway for glucose catabolism. It is two phases of glycolysis. In first phase, the energy requiring stage or investment stage. First, 1 molecule of glucose is converted into 2 molecules of glyceraldehyde 3-phosphate. This step requires 2 molecules of ATP. In stage 2, called energy producing stage, the 2 molecules of glyceraldehyde 3-phosphate are converted into pyruvate, that is aerobic glycolysis or lactate anaerobic glycolysis. These steps produce ATP molecules. You can see in this picture, glucose is converted into glucose 6-phosphate with the help of enzyme hexokinase, and ATP is converted into ADP. ATP means adenosine triphosphate converted into adenosine diphosphate. 1 molecule of ATP is used in this step. Then glucose 6-phosphate is further converted into fructose 6-phosphate with the help of enzyme phosphohexose isomerase, and fructose 6-phosphate is further converted into fructose 1,6-diphosphate, and 1 ATP is utilized in this step with the help of enzyme phosphofructokinase. Then fructose 1,6-diphosphate or bisphosphate with the help of enzyme aldolase is converted into 2 compound dihydroxyacetone 3-phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is further converted into dihydroxyacetone 3-phosphate with the help of phosphotriose enzyme, and dihydroxyacetone 3-phosphate is converted into glyceraldehyde 3-phosphate with the help of isomerase enzyme. Now we can take this glyceraldehyde 3-phosphate, and this glyceraldehyde 3-phosphate is converted into 1,3-diphosphoglycerate and 1 NADH, 1 NAD is reduced to NADH with the help of triphosphate dehydrogenase. Then this 1,3-diphosphogylicerate is converted into 3-phosphoglycerate with the help of enzyme phosphoglyceric acid kinase, and one more ATP is formed. 3-phosphoglycerate is converted into 2-phosphoglycerate with the help of phosphoglycerate mutase enzyme. 2-phosphoglycerate is converted into phosphoenol pyruvate with the help of enzyme enolase. And this phosphoenol pyruvate is finally converted into pyruvate with the help of enzyme pyruvate kinase, and one more ATP is synthesized in this step. You can make this glycolysis cycle with these structures also. And next is stepwise explanation of glycolysis. So first step in glycolysis is called phosphorylation. Glucose is phosphorylated by ATP to form sugar phosphate, and this is irreversible reaction and it is catalyzed by the enzyme hexokinase. The next second step is called isomerization, reversible rearrangement of chemical structure of carbonyl oxygen from carbon 1 to carbon 2, forming a ketose from the aldolase. Thus, isomerization of the aldolase glucose 6-phosphate gives the ketose and fructose 6-phosphate. Step third is also called phosphorylation step, because in this step fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Next step is breakdown. The 6-carbon sugar is cleaved to produce two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by the enzyme aldolase. And fifth step is called isomerization. Dihydroxyacetone phosphate is oxidized to form glyceraldehyde 3-phosphate. This reaction is catalyzed by the enzyme triose phosphate isomerase enzyme. Step 6. Two molecules of glyceraldehyde 3-phosphate are oxidized and converted into 1,3-biphosphoglycerate. Next step is transfer of high energy phosphate group that was generated earlier to ADP form ATP. This phosphorylation, that is addition of phosphate to ADP to give adenosine triphosphate, is termed as substrate level phosphorylation, as the phosphate donor is the substrate 1,3-bisphosphoglycerate. The product of this reaction is two molecules of 3-phosphoglycerate. Step 8. Remaining phosphate ester linkase in 3-phosphoglycerate is moved from carbon 3rd to carbon 2 because of relatively low free energy of hydrolysis to form 2-phosphoglycerate. Ninth step is called dehydration step, in which 2-phosphoglycerate is dehydrated with the help of enolase enzyme and converted into phosphoenolase pyruvate. The compound is the phosphate ester of the enol tautomer of pyruvate. This is reversible reaction. So next is energetics of the glycolase. In first step, glucose to glucose 6-phosphate 1 ATP is utilized, and fructose 6-phosphate to fructose 1,6-bisphosphate another 1 ATP is utilized. In seventh step, 1,3-diphosphoglycerate to 3-phosphoglycerate 2 ATP is synthesized. In phosphoenol pyruvate, another 2 ATP is synthesized and glyceride 3-phosphate converted into 1,3-bisphosphoglycerate. NAD 2 NAD is reduced to 2 NADS, and in 1 NADH 3 ATP is used. So here net ATP synthesized is 8. Significance of glycolysis. Presently, in nearly all living organisms, glucose is the source of almost all energy used by cells. It is the only source of energy in RBCs. Universal central pathway of glucose metabolism occurring in cytoplasm of all the tissues of biological system, leading to generation of energy in the form of ATP of vital activities. It also helps in the synthesis of non-essential amino acid. The next is tricarboxylic acid cycle, that is also called TCA cycle, Krebs cycle. It is a central metabolic pathway. It is discovered by Sir Hans Krebs in 1937. The citric acid cycle is nothing but the forward procedure of the aerobic glycolysis pathway. Essentially, it involves the oxidation of acetyl coenzyme A to carbon dioxide and water. Final common oxidative pathway for carbohydrates, fats, amino acids. The end product of aerobic glycolysis pathway, pyruvate, is converted into acetyl coenzyme A, which start the citric cycle. The most important central pathway connecting almost all the individual metabolic pathways, supplies energy and also provide many intermediates required for the synthesis of amino acid, glucose and heme. The main location of TCA cycle is in mitochondrial matrix in close proximity to the ETC. The citric acid cycle occurs in only aerobic condition. Sorry. The glycolysis occurs in both aerobic condition and anaerobic condition. The citric cycle occurs in only aerobic condition. The first step of TCA cycle, that is conversion of pyruvate to acetyl coenzyme A. You can see in this structure, this pyruvate molecule convert to acetyl coenzyme A, where NAD is reduced in NADH and CO2 released with the help of coenzyme A. In first step, pyruvate is converted into acetyl coenzyme A with the help of this enzyme, and then acetyl coenzyme is acetyl coenzyme A condensed with oxaloacetate and convert into citrate. Then this citrate is converted into isocitrate and with the help of aconitase enzyme. This isocitrate is converted into alpha ketoglutarate with the help of enzyme isocitrate dehydrogenase, and NAD is reduced in NADH. Then alpha ketoglutarate is converted into succinyl coenzyme A with the help of enzyme alpha ketoglutarate dehydrogenase. Then this succinyl coenzyme A is further converted into succinate, and 1 mole of GTP is synthesized, and enzyme is succinyl coenzyme A synthetase. This succinate is converted into fumarate, and FAD is reduced to FADH2 with the help of enzyme succinate dehydrogenase. Then fumarate with the help of fumarase enzyme is converted into malate, and then further malate is converted into oxaloacetate with the help of malate dehydrogenase enzyme, and NAD is reduced to NADH. Then NADH and FADH2 are the coenzymes which are used in the synthesis of fatty acids. Next is reactions of TCA cycle. First is formation of citrate. Oxaloacetate condensed with acetyl coenzyme A to form citrate, catalyzed by the enzyme citrate synthase, inhibited by ATP, NADH citrate, competitive inhibitor of oxaloacetate. The first step in this is the formation of citrate. Second step and third is citrate is isomerized to isocitrate. And step fourth and fifth, formation of ketoglutarate. And step 6 is conversion of alpha ketoglutarate to succinyl coenzyme A. Step 7, formation of succinate, substrate level phosphorylation. Step 8, conversion of succinate to fumarate. And step 9, formation of malate. And step 8 is malate dehydrogenase. Malate is oxidized to form oxaloacetate. Step 10th is conversion of malate to oxaloacetate. So energetics of TCA cycle. Oxidation of 4-NADH by ETC coupled with oxidative phosphorylation result in the synthesis of 12 ATP. FeDH2 leads to the formation of 2 ATP and 1 GTP is equal to 1 ATP. One substrate level phosphorylation, thus a total of 15 ATP are produced from one acetyl coenzyme A. Since here are two molecules of pyruvate or acetyl coenzyme A, hence the total ATP formed in Krebs cycle is 15 into 2 is equal to 30. So total ATP from one glucose is 8 and from Krebs cycle is 30. So net ATP is 38 ATP. Significance of TCA cycle, complete oxidation of acetyl coenzyme A, ATP generation, final common oxidative pathway, integration of major metabolic pathway, excess carbohydrates are converted as a neutral fat, no net synthesis of carbohydrates from fat, carbon skeleton of amino acids finally enter the TCA cycle. So next is gluconeogenesis. Gluco means glucose, neo means new, and genesis means formation. So synthesis of glucose from non-carbohydrate compound is known as gluconeogenesis. Gluconeogenesis is not a common process and occurs only during prolonged fasting and starvation and intense exercise. The major substances precursors are lactate, pyruvate, glucogenic amino acids, glycerol. It is energy consuming process. Location mainly cytosol, although some precursors are produced in mitochondria, mostly take place in liver and to some extent in kidney matrix. So reaction of gluconeogenesis, it is closely resemble the reverse pathway of glycolysis, although it is not the complete reversal of glycolysis. Essentially, 3 out of 10 reactions of gluconeogenesis are irreversible. 7 reactions are common for both glycolysis and gluconeogenesis. 3 sorry, the irreversible step of glycolysis are catalyzed by the enzymes, namely hexokinase, phosphofructokinase, and pyruvate kinase. These three stages bypassed by alternate enzymes specific to gluconeogenesis are discussed. So first step is conversion of pyruvate to phosphoenol pyruvate. This takes place in two steps. Pyruvate carboxylase is a biotin-dependent mitochondrial enzyme that converts pyruvate to oxaloacetate in the presence of ATP and CO2. This enzyme regulates gluconeogenesis and requires acetyl coenzyme A for its activity. Conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. Phosphoenol pyruvate undergoes the reversal of glycolysis until fructose 1,6-bisphosphate is produced. Third step is conversion of glucose 6-phosphate to glucose. Glucose 6-phosphate catalyze the conversion of glucose 6-phosphate to glucose. The presence or absence of this enzyme in a tissue determines whether the tissue is capable of contributing glucose to the blood or not. It is mostly present in liver and kidney, but absent in muscle, brain, and adipose tissue. So this is the gluconeogenesis, that is reversible of glycolysis. In glycolysis, we produce glucose to pyruvate. Glucose is converted into two molecules of pyruvate, but in gluconeogenesis, pyruvate is converted in glucose. So pyruvate is first converted in oxaloacetate with the help of pyruvate carboxylase enzyme. Then it is converted into phosphoenol pyruvate with the help of phosphoenol pyruvate carboxykinase enzymes. Then this phosphoenol pyruvate is converted into 2-phosphoglycerate with the help of enolase enzyme. This 2-phosphoglycerate is converted into 3-phosphoglycerate. Then 3-phosphoglycerate is converted into 1,2-bisphosphoglycerate, and 1,2-bisphosphoglycerate is converted into glyceraldehyde 3-phosphate, and glyceraldehyde 3-phosphate is further converted into dihydroxyacetone phosphate with the help of triose phosphate isomerase enzyme. This dihydroxyacetone phosphate is converted into fructose 1,6-biphosphate with the help of aldolase enzyme, and this fructose 1,6-biphosphate is converted into fructose 6-phosphate, and fructose 6-phosphate is converted into glucose 6-phosphate, and last glucose 6-phosphate gives glucose with the help of glucose 6-phosphatase enzyme. So significance of gluconeogenesis is to provide glucose for the production of energy when sufficient carbohydrates are not available for the energy production. It happens when our body is witnessing a lack of stored glucose in the form of glycogen in the liver and muscles. Ideal to get glucose in the system when a person is suffering from diabetic condition, doing heavy exercise or fasting. Ensures a continuous supply of the glucose for erythrocyte and the nervous system. It maintains blood glucose level when required. It is used to clear metabolic products of other tissues from blood such as lactate and glycerols. Thank you.